Hello, good afternoon. My name is Juan Morales. I am a DOE fellow, and I would like to talk to you about research that I've been conducting at the Applied Research Center in Florida International University. I am a PhD candidate in the Department of Environmental Health Sciences, and the title of my presentation is called Modeling Episodic Heavy Metal Transport and Toxicity for the Assessment of Remediated Surface Waters. Touching base on a brief background, I want to talk to you about the introduction of my research, the materials and methods, results, and lastly, some of the conclusions. So looking at the hydrological cycle, of course, many of us are familiar with the concept of how water moves through the system. It can be through urban or rural areas, but I'd like to focus on how contaminants move and how they behave in the system. It can be pesticides, fuels, heavy metals, organic compounds, or nutrients. But on a watershed scale, you can really have an expansive aquifer. You can have tributaries and rivers, and each of those can receive contamination or become contaminated. So how do you model and assess the movement of those contaminants? More importantly, how do you set up or how do you forecast the impact regarding the management practice and mitigating the spread of pollution has been my area of interest. So some background on the environmental impact of heavy metals. Heavy metals, by definition, are naturally occurring elements that have a high atomic weight and a density at least 5 times greater than of water. Among many of those pollutants, heavy metals have received great attention given the toxicity of their nature. They're present in trace amounts in natural waters, and many of them can become toxic even at low concentrations. So as they build up, they can become a significant health hazard. I borrowed this quote from CERCLA's risk assessment portfolio. So remediation of complex environments, it's by definition, it's reducing, removing exposure from contaminated soil, groundwater, surface water by the application of technology in order to help regulate, to meet regulatory requirements. However, long-term monitoring is associated with the effectiveness of reducing the risk. There are a lot of models, and models are considered to be very essential in an essential part, and they should be proportional to the size of the project. EPA, however, mandates that remediation and containment strategies should be strengthened in order to reflect the changes of climate change. So which brings me to my case study research area. Here I've highlighted Savannah River site. This is the 300 square mile facility. It's owned by the Department of Energy. It is located in Aiken, South Carolina. And the history behind Savannah River is that during the World War II Manhattan Project, large volumes of nuclear waste were generated. So it was considered to be one of the major processing facilities during the Cold War where plutonium was enriched. There are many branches in DOE, and I'm currently conducting research for the DOE's Office of Environmental Management. So the SRS-AM area, I've mentioned before, it was a nuclear fuel processing facilities. And there has been a lot of contamination in that watershed. So there has been discharges recorded in which 43,500 kilograms of depleted uranium have entered the Timms Branch water system, along with much of the uranium. Wastewater containing other metals and radionuclides such as aluminum, nickel, manganese, mercury, lead, copper, chromium, and zinc were reported as a result of those discharges. And studies suggest that an estimated 70% of all of those metals have been sequestered in the stream of the sediment, mostly in Speed Pond. the history behind the AM area, along with all of that contamination, in 2007, remediation technology started, and within remediation, an air stripping technology in which stannous chloride is injected into the groundwater and then aerated in order to reduce mercury was installed in 2007. This effectively removed about 90% of the mercury concentrations. And most importantly, I'm interested in anything that feeds the Thames Branch water stream, given that the majority of the stream flow from Thames Branch is being fed by this groundwater pumping. So what does long-term monitoring for the Savannah site mean? So the site needs not just SRS, but other DOE sites are challenged with heavy metals and radionuclide contamination, both in the subsurface and surface environments. Many of these sites are pushing towards site closure. So given that they want to give it back to the community and to move forward with site closure, they would like to evaluate the effectiveness on the applied remediation technologies. Most importantly, they're interested in establishing a long-term monitoring strategy. So here I am highlighting two objectives in which I plan to research. The first one is to develop a contaminant transport model and to temporarily simulate discharges of heavy metal flux as a result of increased precipitation events. I am looking at 5 to 500 year adverse recurrent intervals. Most importantly, in that objective, I would like to evaluate the percentage of uranium transported downstream of Steed Pond and at the outlet of Thames Branch Stream compared to base flow conditions. My second objective is to use information from the model regarding several concentrations at the different areas in the stream. So I want to develop a zebrafish gene environment network discovery model in order to integrate using integrated systems biology to model hazard assessment and to improve the knowledge about the remediation benchmark doses already established at Savannah River site. Looking at the overall schematic of the contaminant transport model, there's just like any model, there's a lot of input variables in which they need to be inserted in order for the model to be representative of the research area. With those, we have topography, climatic drivers, time series data, land cover data, soil types, all different cross-sectional profiles. And also we have, I have installed several monitoring stations in which I am collecting water level and turbidity information. All of this information is stored into a geodatabase, and then with the help of DHI software models called MyShe, Mike11, and Ecolab, I plan to accomplish this objective. So here we have, we go from the DHI MyShe model into the sensitivity analysis and calibration, looking at how I've designed storm events, and I am going to show how I've executed a water quality model for this watershed. So touching a little bit more in detail about the model input, this slide outlines the model sources and the inputs used currently. So for the model development, I have primarily used data sources and layers for topography, time series for climatic drivers, and such as evapotranspiration. To make sure the model was representative of the research area, we used different land data types and vegetation types. For example, here I've highlighted that we're using 15 different vegetation types, such as root depth and leaf area index. The same with the soil type. As an example, there are at least 44 different soil types, each of them describing and associated with their soil physical properties, such as hydraulic conductivity versus the water content. So for the model calibration, we primarily used stream flow discharge point, and this was provided by the monitoring stations I have mentioned previously. However, most of the data was available from federal and government databases, as highlighted here in this slide. We have conducted some field sampling in which this information has been documented, such as uranium concentrations, heavy metals concentrations, and this map highlights all of the areas that were sampled. To further continue with the application and equipment deployment, as you can see here in the picture highlighted on the left, several devices were installed in the stream and cross-sectional profiling were documented and inputted accordingly to the model. Following the model development, we ran a sensitivity analysis to identify a number of model inputs that are more influential to the outputs. So after the model was calibrated, the model was able to predict very closely to the observed data as described here by this hydrograph. At the bottom of the figure, as you can see, is represented in a bar plot. This identifies precipitation distributed by time and magnitude. Meanwhile, the blue line represents the stream flow, or others might like to call it observed data. The red line represents the predicted or simulated data values. After achieving a reliable hydrological model, we began building the model capacity to predict the metal transport. In this case, this was done by coupling Mike 11 and Mike Ecolab module. Mike Ecolab module here, stated on the picture, this simulates sediment transport to the through sedimentation and resuspension, as well as the interactions with heavy metals through adsorption, desorption, and diffusive transfers. Regarding the event-based design, we prepared the model domain in order for the analysis. So we integrated both models, Mike Xi and Mike 11, and we had to recalibrate it to different groundwater levels and flows were centered to the outlet of the watershed. Parameters were optimized and were based on selected through literature review and hydrology modeling standards. So, for example, we redigitized the streams using 1 meter digital elevation model, and cross sections were made wider to improve the conveyance for the drainage of the extreme flows. The horizontal conductivity band was assigned using the auto calibration process, and this was then executed using the model server, in which about 1,200 runs were combined in order to obtain the most influential parameter for the model. So in order to simulate for the extreme events, we chose a 1 to 500 years and performed an analysis in which different spreadsheets with time series were developed. This is some results regarding the forcing results for the Mike 11. So to assess the significance of the extreme events in the transport of contaminants, a period of 1 to 500 years was considered. Here I'm highlighting about 192 flow hydrographs in which the Mike 11 was able to model. The area of interest here, as you can see, they're both custom selected. One of them is the one on the left is below Steed Pond, and the one on the right is the outlet of the Timms Branch outlet. So they describe an area in which Steed Pond and Timms Branch outlet are of our research interest regarding uranium transport and heavy metal transport. Regarding the calibration results for the water quality model, we simulated a different series of exercises in which we're highlighting organic carbon partition coefficient to be a major driver in uranium flux at the outlet of the TIMS branch. We found out regarding our observed data, we used Batten et al. Regarding validating our uranium transport model. So we determined that the uranium flux fell in agreement with the observed data reported by that team. Finally, here I'm highlighting different forcings in which 5 to 500 year events are highlighted on the left. Meanwhile, the picture on the right, the figure on the right, highlights the uranium flux as a function of time and as a function of forcing. With this, we're able to quantify that the greatest amount of uranium was transported during the 500-year ARI, peak discharge rate. Importantly, and more interesting, considerably more uranium was transported to the TIMS branch outlet. As a result, about 3,550% increase for the storm simulated, and 1,327% increase in Steep Pond when compared these uranium flux measurements with the base flow measurements. Also, we identified that the uranium transport is governed by organic carbon partition coefficient, for which field and literature sources were used in this exercise. Lastly, the transport of uranium sorbs to soils could be underestimated when calculations with further uranium flux are based on base flow measurements. Regarding my second objective, the Gene Environment Network Discovery Model, I am using zebrafish transcriptomics. Zebrafish are a well-known model in which are used as sentinels for environmental toxicology. So zebrafish transcriptomics and gene expression can be used for markers in the development and regarding vascularization, neurogenesis, cardiotoxicity, hepatotoxicity, and other biological pathways regarding environmental contaminants. So it's important because transcriptomics are really good regarding that they account for the complete RNA content of the cell. They can be used at a single point in time in which concentration can be looked into in different time points. And we are able to compare between different organisms such as humans and mice and other species. My design uses a combined ChIP-seq and DNA microarray in which I'm downloading data sets from public repositories. I'm also using a redox sensitive transcription factor to be well known in the realm of as an environmental sensitive transcription factor. Which leads me to nuclear respiratory factor 1. Is a well-established transcription factor and is known for the role in activating the expression of many target genes, most importantly involved in mitobiogenesis, DNA replication, and respiration. Genome-wide association studies have also looked into NRF1 regarding cell cycle regulation. Also with other species, NRF1 is found in humans, zebrafish, mice, and Drosophila. I've mentioned before that this is a very well-documented transcription factor and is stress sensitive, which I am trying to use as a marker for heavy metal exposures. Regarding the data that I'm using, this is the demographic table I've created. So several compounds are listed in the left column, going all the way from nickel, chromium, cobalt, methyl mercury, all the way down to lead, copper. So in order for you to understand how this data has been classified, here I am looking into different tissue types in which embryo, whole body dorsal fin, and hepatocytes have been classified independently. We have different concentrations in which I've categorized them as low, medium, and high. and I'm using different accession numbers with different GPL platforms. All of these are CDNA microarrays. Regarding the workflow of how I'm analyzing the data, I've mentioned before that I have embryo, liver, and muscle cDNA microarrays in which data has been extracted, processed, and normalized. I've also standardized all of the data using Z-score. I am looking into identifying the differentially expressed genes across platforms, and I am using a false discovery rate of 0.05, and that's in order to account for batch effects. The candidates of interest have been selected using fold change plus or minus 1.5 and p-value of 0.05. I've also made a little bit of stringent criteria, and I've done a correlation ranking in which the majority of the genes were ranked according from gene expression as a function of heavy metal. Looking at the NRF1 targets, I've cross-referenced all of my targets using a ChIP-seq database called ENCODE. Regarding the buildup of the gene environment network discovery, here I am classifying the data into embryo, liver, and muscle. The variables of interest in which the machine learning aspect takes into consideration. You need to discretize all your variables into 0, 1s, and 2s, and I've chosen dose, gender, and tissue specificity as a variable of interest. The model I'm using Banjo is called Bayesian models with Java objects, in which two criteria have been selected. One of them is a Markov blanket or constraint-based algorithm, and the other one is BDE scores, in which the score is based, I am determining based on the best score which structure I'm selecting. Regarding the model interpretation and validation, I am using several platforms in which to create heat maps, go-term evaluation, and pathway analysis. Lastly, for the environmental health environmental health risk evaluation will be assessed. So the raw data was processed using the raw TIFF files in which the image was processed and transformed and normalized. I have normalized all of the data using the total intensity normalization, and the relative ratios were converted into expression ratios. This information was corrected by subtracting the signal noise to the ratios. As you can see here, we're only extracting the data from the inside of the circle, not the outside. We're subtracting this information. Here, this highlights all of the data has been normalized. For the sake of this experiment, my preliminary analysis shows regarding the metal hepatocytes as a contaminant compared with the control samples. We have several up and down regulated genes. Several of these genes were mostly identified in the 48-hour treatment. I've mentioned before that the presence of batch effects were reduced because I integrated the false discovery method. And I've compared controls versus treatment, and I've selected the criteria cutoff of p-value 0.05 were selected in this exercise. Regarding the identification of the NRF1 markers, I've used volcano plots in order to highlight which genes were significant in order to have a two-set criteria, which is 0.05 and fold change of 1. As you can see, all of the genes highlighted in red will be used for this exercise. Now, when building the network structure learning using hybrid algorithms, I've mentioned before that we used the Bayesian analysis with Java objects, and in which all of the NRF1 targets were introduced as discretized variables. So I've used, this is the design of the model, in which 0, 1, and 2, we have 3 sample replicates, and the machine learning sampling was introduced as 1, 2, 4, and 8 hours of runtime. We have 8 hour, 24 hour, 48 hour, 96 hours, and the best structure from this category was introduced into the 2-hour component, and the best structure from this 2-hour component was introduced into the 4-hour, making and choosing the best network in the overall simulation. We did the same thing for 8, 24, 48, and 96 hours. Now, we used the model's output information, and the best networks were selected. Here, I am highlighting the first order market blanket genes as identified using Cytoscape. And a heat map was generated in order to show the normalized gene expression as a function of arsenic stress, and this is temporally visualized. This is very interesting to see. We see that several NRF1 markers were significantly downregulated at 48-hour treatment time. Using the markers already modeled using machine learning, we use gene ontology as a bioinformatics platform in order to identify the molecular pathways identified and compared from curated data. So here, gene ontology highlights the increase in annotations as a result of arsenic treatment and the relationship of groups of genes being enriched. So this is a very informative platform in which using the biological regulation parameter, we can observe that at the 48 hours of exposure, the greatest amounts of cascading changes were as a response of stress. Lastly, I want to conclude by saying that the supervised machine learning model improved the runtime. The optimal network was achieved at 8, 24, 48, and 96 hours of treatment, and was at 8, 2, 1, and 2 hours of simulation runtime. Using Banjo's hybrid algorithm, we identify 4 key NRF1 molecular signatures. These are influential to the arsenic treatment in zebrafish hepatocytes, PSMA2, CTSD, SMARC, and MTS1. The gene expression was detected among PSMN2, CTSD, and MST1, in which resulted in increased expression driving biological regulation. Regarding the pathway analysis, this showed that NRF1 targets played a key role in the cell cycle, NRA transport, P53 signaling, and glycol serophospholipid biosynthesis in the zebrafish hepatocytes. Lastly, this molecular framework has proven to be very effective in identifying the NRF1 stressor gene signatures, and more importantly, this can aid in the environmental monitoring as an alternative to chemical testing in the aquatic environments. I want to wrap up and acknowledge all of my committee members, my major professor, co-major professor, my committee members, and my collaborators at Savannah River National Lab, Brian Looney, Dr. Dan Kaplan, and my program director, Dr. Leona Lagos, Peter Heisenberg, the postdoc associate, and Angelique Lawrence, the GIS specialist. Thank you very much, and have a great day.